it happens, it becomes difficult to correct it down the line. So that is why it is essential to improve cassava. Next slide. So another reason why it is important to improve cassava is because of the bottlenecks that is being experienced in breeding. Breeders have done a lot to improve cassava, like what we have now to improve cassava with good yield, with good dry matter content, with vitamin A and other things that we need. But there are bottlenecks. For example, long breeding cycle. It takes approximately 8 to 15 years for a breeder to come up using the conventional method to come up with a new variety. You will agree with me that anything that is out there in the environment depreciates. Even your car depreciates, your house depreciates. So even the variety that you produce today that is good depreciates over time. So there is need to keep developing new varieties that will replace the old ones. So some of these setbacks, we have low seed set. For example, when you have a parent, a male and a female, you cross them to get offspring seeds that you will eventually evaluate and develop as a variety. Most times in cassava, you have very low seed set, insufficient number of seed that comes after the breeding. So that's a bottleneck. Another thing is there is this phenomenon we call heterozygosity. And what it means is that, for example, in every characteristics or trait in a living organism occurs as a pair. And when the pair is the same, it helps in breeding, facilitating breeding. But when it is different, for example, in the diagram you see here, in the homozygote, you have B, 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 B. And then in the heterozygote, you have B and A. So when you have such things occurring, it makes breeding a little difficult. Also, in cassava, we have lots of genes that are not really relevant in the things that we desire it to do. So when you cross, you're transferring the preferable traits and non-preferable traits. We call it high genetic load. So this is also what makes breeding difficult in cassava. Then we also have another phenomenon we call high polymorphism. What we mean is that variation in appearance. There's so many variation in appearance in cassava. So some of these things affect breeding in cassava. So next slide. So what are the current improvement strategies? All right. Okay. So the hybridization breeding, where you have a male and a female, and then you do crosses to get offspring. Then we now have other innovative technology in breeding, like genome selection, marker-assisted selection, transgenic breeding, and of course, the genome editing that is our main focus for today. So for want of time, I'm not really going to go into describing these other improvement strategies. So I'm going to focus on genome editing and what we're doing with it. Next slide. So what is genome editing? Or some people also call it gene editing. So what that means is simply rewriting the genetic information of living organisms. For what purpose? To create new abilities, correct abnormalities, and to improve performance. I repeat, I said, it is rewriting. Now, like what you see on the screen, that's my pencil and that's my eraser. So I am rewriting the genetic information that determines the characteristics of an organism. I'm trying to rewrite it so as to create new ability, make the organism capable of carrying out new functions, and then correct abnormalities that could have resulted as a result of the organism living in a particular environment, and as well as to improve the performance of the organism. Genetic information controls the characteristics of an organism. For example, the way you are, your appearance, tall, fair, plumpy, and the things you can do, the way you talk, the way you act, all these things are determined by genetic information that is carried in your cell. All living things, bacteria, animal, plants, human beings. Now, I have an example on the screen. Let's take, for example, the red blood cell, the blood disease. So we have two types of red blood cell. We have the normal red blood cell and we have the sickled red blood cell. Now, for the normal red blood cell, if you see on the screen, there is a genetic information there of physical characteristics of the blood cell. Now, by reason of mutation, by mutation, we mean an inheritable change that occurs. Mutation occurs in life. There are a lot of things that can cause mutation. It can be physical agent, it can become chemical agent, it can also be biological agent. So anything can cause mutation. So by reason of mutation, there is a change in the genetic information. The GAG now becomes GTG. And when that happens, it produces a phenotype, a physical characteristics of a sickled red blood cell that is deficient in carrying out a proper function of distributing oxygen in the body. So genome editing, what do? We try to discover, find out the genetic information that controls a particular trait or characteristics. And then if there is a defect in that genetic information, we can use genome editing tool to make a correction. For the example we see now, we use genome editing tool to change the T that is in the sickled cell back to A. And when that happens, you restore the original red blood cell. So that is the principle of genome editing. So next slide. So what are the tools? What are the things that we're actually using to do this? So there are different kinds of tools, but the basic ones that are being used are these four. One is called the mega nuclease. Two is called the zinc finger nuclease. Three is called the TALIN. And the fourth is called the CRISPR-Cas9. They are different in some levels, but they are characterized by mainly the same features. So what are these features? These tools have a portion that recognizes a target. For the example I use, the GTG is our target. The T is what we want to change. So these tools have a recognition portion that helps them identify the T. And then they have a cutting portion that helps to cut out the T. So these are the characteristics of this genome editing tool. So next slide. So what is the mechanism? Now, let me back up a little bit. I will use CRISPR-Cas9 for our discussion today. Now, CRISPR-Cas9 is a mechanism that was naturally discovered in bacteria. Now, bacteria are usually affected by viruses that we call bacteriophage. So when the bacteriophage infects the bacteria, it takes over the cell, it takes over the bacterial system, and instead of the bacteria producing its own cells, it begins to produce the virus cells, and thereby it takes over and kills the bacteria. So nature has a way of protecting itself. What bacteria does is that it has this mechanism, this system innate, which is the CRISPR system, that helps it recognize the invasion of viruses and then uses that portion that cuts it like a scissors to cut short, to cut in pieces the viruses, thereby preventing the viruses from taking over. It's just like in human beings, we take vaccines. So what the vaccine does is to introduce a portion, a safe portion of the organism that causes disease and makes our body to produce antigen that will help us fight the disease. So this is where CRISPR-Cas9 came from bacteria. So scientists are now using this mechanism to help us edit genetic information in living things. Remember I said, the portion, it has a portion that recognizes a sequence. Then it has a portion, like from what you're seeing on the screen, you see the sequence that's called sgRNA, helps to recognize a sequence. Then you see the scissors. The scissors cuts. So the diagram below now, you see there is a cleavage, there is a cut. So that is where, if this is our sickle cell gene now, it means that we have cut off the T. So the body has a repair mechanism, like double-stranded break cuts in our DNA. It happens all the time, both in plants, in human, in animal. When it happens, nature has a way of repairing the double-stranded break. So we now use this tool to cause a break in an organism, and then also allows the system to repair itself. So that's genome editing, the mechanism. Next slide. What is the difference between genetic engineering and genome editing? So in genetic engineering, the basic difference are this: genes are introduced from outside the organism. So what it means is that I want to improve, for instance, I want to make bacteria to produce insulin, and bacteria doesn't have the mechanism to do that. So I go to human beings, isolate, get the gene that is responsible for producing insulin, and I put it in bacteria to enable the bacteria to produce insulin. That is genetic engineering. That is what we call transgenic, that is transgene. You're taking a gene from one organism to the other. And the integration can be anywhere in the genome. That's for genetic engineering. Now for genome editing, what we do is you cut and repair a sequence, as I've explained the other time, and the editing is at a specific or targeted site. Next slide. So how are we applying this in cassava? So what we're doing is that we're using genome editing for increased understanding of the role of sweet gene in cassava bacterial blight disease. Remember, I said that one of the reasons why we're improving cassava is because of the impact of disease. So one of such disease is cassava bacterial blight disease. If you look at the screen, the picture at the upper part of the slide, you see a healthy cassava field in Uganda. Then the lower picture is this particular field, the same field, affected by CBB. So the important thing about this disease is that we don't even understand the trend. What I mean is that we don't know what happens, what makes it happen, why does it occur? For instance, last year, most of our fields were impacted by CBB, and you will come this year, you might not see the same effect. So we've not really grasped the mechanism, the trend of the disease. So with genome editing, we are trying to gain this understanding. How does this happen? And how can we take care of this? So the causative agent for this cassava bacterial blight is what we call Xanthomonas azonopodes PV manihotis, for short ZAM. What it does is that the bacteria attaches itself to a plant cell, to cassava. And then this bacteria releases its own ammunition in forms of protein that goes inside the cassava cell and forces the cassava to begin to produce sugar. This sugar helps the bacteria to grow very fast, colonize the plant, and then cause disease. So our thinking, this gene that is colonized by the bacteria is what we call susceptibility gene. This is the gene that makes cassava susceptible, that makes cassava prone to this disease. So our thinking is this: if we could identify this gene and use genome editing tool to remove this gene, because our previous studies have really shown that this susceptibility gene is not doing so much for the bacterial for cassava growth. So we can kind of remove it, cut it, manipulate it to see if that happens. Then the bacteria will be disarmed. It will not be able to grow massively and to cause disease. So next slide. What is our goal? Our goal is to cut off the portion of that gene that enables the bacteria to multiply and to grow. So from the diagram you're seeing, you see the orange, not the yellow portion. So that yellow portion is the genetic information that we're targeting. We believe that when we knock this out, the terminology we use there is knockout. That means we want to break it. We want to use the genome editing tool to remove one or two genetic information there and thereby disrupt it from being expressed, from being used by the bacteria. So that is our first target. Then our second target, can you move down a little bit, the slide? Can you scroll? Okay, so our second goal is to use the genome editing tool to also develop a disease monitoring tool. Remember I said, we don't know so much about this, the disease progression. So we're thinking we could use this genome editing tool to develop a disease model that can help us understand the progression of this disease in cassava. Next slide. So what did we do? So first and foremost, we designed the gene editing tool. I'm not going to bother you with how we did it, but that is it. That is the tool that we used. So it has the recognition portion and it has the cutting portion. That is the important thing for us to know here. And then after developing it, we have to deliver it into cassava. Now, for example, going back to the example, the sickle cell example I used, for instance, if I'm a sickler and I want to use gene therapy to correct that abnormality, so you cannot just inject the genome editing tool into me to begin to do the correction. So you need to get a tissue from me. In medicine, it's called the stem cells, a tissue that can allow you manipulate, transfer the genome editing reagents into the cell, and after which you put it back in my body. The same thing applies in cassava. So we collect cassava from the field, we establish it in the lab in tissue culture, then we take a portion, a tissue, a very young tissue from cassava to make cells that we can manipulate easily. So this cell is what you react with the genome editing reagents. So after the reaction, you now select those cells that actually received the gene editing reagents, and then you regenerate your cassava back again for evaluation in the field. So that is what we did. Next slide. So our results for the first goal. Now, after reacting the genome editing reagents with the cassava, so we need to test to find out if actually these cells have the editing, the changes that we intended to make. We use a molecular tool that is called polymerase chain reaction, for short PCR. So this tool enables us to go into the cell and bring out, amplify, bring out that portion that we were targeting to change. For us, remember I told you is the yellow portion. So we use this tool, amplify it out, bring it out from the cell, and then to see if the change that we're intending to make was there. We also use what we call sequencing, like the result you see on the right side, whereby you develop the genetic information again to see if you have made the changes that you desired. Now, looking at the right side, the sequence that you see on the screen, you can see the areas where you have dash, 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 dash. These areas, these are the areas where the genes were knocked out. So yes, we achieved the knockout with the gene editing tool, the CRISPR-Cas9. So you could see these are the two lines that were developed from these experiments. So the next step now is that these two lines that were developed are now being multiplied in the lab for testing for CBB resistance. Next slide. So the second goal of developing a disease model. So what we did is this. We tried to use what is called a reporter gene. What it means is that something that can give you a signal if it is present. Here, it is called green fluorescent protein. If it is present in an organism, when you excite it, you will see a signal. So we tried to use this fluorescent protein and attach it to this susceptibility gene, which is the sweet gene that is responsible for bacterial multiplication. We attached it to that gene so that when we induce it, when we trigger it, the fluorescent protein will show, the signal will show. Then we can monitor how the disease is moving. Does it come from the leaf? Does it come from the roots? Does it come from the shoot? Where does it go to? How does it establish? So that was our aim in doing this. So if you could scroll a little bit down, you could see the pictures. Can you go? Yeah. So you see the picture on the screen. So the control is one that doesn't have that was not edited, but the one where you see green was the one that was edited. And then you could see, we also use the same molecular tool, the PCR, to amplify the targeted sequence to see if we had made a change. So now this is being developed. It's being developed as a disease model for understanding CBB progression in cassava. So next slide. So conclusion. So far, what we have done so far, we can comfortably use this genome editing to CRISPR-Cas9 to make any deletion, to make any intended change in cassava. Whichever target that you want to change, we could effectively use it. And we can also include any signal that we want to attach to help us study whatever effect we want to study in cassava. So I want to say that this is really a way forward for cassava improvement, because it means that there is a lot of possibilities, things that were not initially possible with conventional breeding or with genetic engineering can actually be done using genome editing. So it's really a way forward for cassava transformation. So, and then we are hoping to encourage policymakers to look into, to understand the importance and the benefits of this technology and provide a conducive environment for its effective use, and also to provide funding for effective research in this area. So next slide. I think I've come to the end of my presentation. Next slide. I would like to acknowledge the following. So I would like to say that this work was done in collaboration with the Donald Dunford Plant Science Center with Rebecca Batts, specialist that works on cassava diseases, and then with Nigel Taylor's lab, also that does genome editing. So I also want to acknowledge Award that sponsored this research during my advanced science training, and of course, NRCRI for giving me the opportunity to do this work. Thank you for listening.